It's a pleasure to be here today. Appreciate the opportunity to come and speak to this group. Many things that I have to say are not dissimilar to what Alan has introduced, and tomorrow we have the opportunity to hear from Robert Plenge. So I think you're going to hear kind of three complementary, if somewhat different takes on the relative value and applications of genetics in drug discovery. I was going to skip over this once Alan started, but since his slide didn't come up, I'll spend just a moment on this. So what is the evidence that genetics could be useful from the vantage point of prioritizing the targets that we decide to take forward and the drugs that we develop in the clinic? So just to restate the problem succinctly, over the last 20 years, the cost of drug development has gone up tremendously, over twofold. Latest estimates indicate that the average R&D spend for every drug that is approved is approximately $2.5, $2.6 billion. Now, estimates for those range between $1 billion and 2.6, depending on how you load it. But no matter how you cut it, it's very, very expensive. And as Alan highlighted, a big driver for that expense is the high failure rate that we see in the clinic. Most of our R&D spend happens once we bring a drug into the clinic, especially for larger phase 2 and then large phase 3 clinical studies. And we know, is shown here in the upper right-hand corner of this figure, that the number one reason for failure in phase 2 and in phase 3 is lack of efficacy, with the secondary reason being due to safety risks, hazards that are identified after the drug is already in the clinic. So there have been a number of lines of evidence that have led us to be encouraged that genetics, and really the genetic revolution that we've seen happening over the past 12 years or so, will help guide us to better choices for drug targets that we could develop drugs for. So some of the first lines of evidence were a bit anecdotal. Robert Plenge had a fantastic paper in 2014 where he and David Altshuler went through several examples, which were nice anecdotes to demonstrate where you could potentially leverage genetic information to make better early drug discovery choices. That same year, there was a publication by AstraZeneca, Cook et al., where they had gone back and done a post-mortem analysis of their pipeline over the previous decade or so. And they had looked into what were the variables that differentiated between their projects that failed in early development versus those that went on to be successful. And they showed that there was some evidence that the projects that had some amount of genetic evidence, which was undefined in that paper, but projects that had genetic evidence supporting the therapeutic hypothesis were roughly 3.5 times more likely to be still active programs at AstraZeneca compared to those that lacked that genetic evidence. And so with a desire to try and go back and take a look at this in a more systematic way, we carried out a study where we went back and we leveraged a commercially available data set, pharma projects, that essentially catalogs roughly 73,000 molecules that have been through preclinical and clinical development over the past 55 years or so, and providing essentially what indications were those molecules developed or attempted development for, and what were the outcomes of those studies. And so we partitioned that down and we reduced it down to unique combinations of targets and indications. And we asked for every target and indication that was investigated either preclinically or clinically, how far did any drug for that target indication make it through the drug discovery and drug development pipeline? And then we asked, using the available GWAS data at the time, so this was 2014, a somewhat smaller data set than we have available to us today, which of those target indication pairs are supported by genetic evidence. That is, that we find a genetic association that we can reasonably link to the same gene for a trait that is relatively similar to the indication that went into drug development. And the results of that are shown in the lower right-hand corner of this, where we see that the further that a target indication was able to survive through the drug discovery and drug development gauntlet, the more likely that target indication had the support of human genetic evidence. We were able to use these estimates to be able to estimate that the probability of a drug entering the clinic to survive as an approved medicine at the other end, it was twice as likely to be successful if it had supportive genetic evidence versus those that lacked that evidence. We've updated this analysis in more recent years with the growing body of data, and while we see essentially these graphs shifting to the right, approximately 2.5-fold over what we saw in 2014, the overall conclusion remains the same. We're estimating about a 2-fold increased risk of, or increased probability of success for those that have supporting genetic evidence versus those that don't. So how do we go then from a concept for a new drug, an area that we want to treat, an area of unmet medical need that needs new therapies, to identifying a target that we can develop a molecule to either activate or inhibit and have reasonable confidence in that therapeutic hypothesis to actually have a positive effect in patients? So I'm going to break this down in a somewhat different way and take you at least partway through the journey that we go on from early genetic association through to early decision making in selecting a new drug target. I'm really going to focus on the first three of these, the task of finding a genetic association, trying to understand the impact that a variant that we've identified has across a wide range of phenotypes, and then ultimately trying to establish confidence that we're identifying the right gene and we understand the effects of that gene. Now, there are other steps there that include both in vitro and other drug discovery activities that I'm not planning on going into in the talk today. So let's start with step one. So finding the genetic association. So all of us here probably are very well aware of kind of where we've been and how far we've come in just the last 12 years. Where 12 years ago we could literally count on two hands the number of confirmed genetic associations for common complex traits. And now if you look at the EBI GWAS catalog, and this is as of just a few months ago, there are nearly 30,000 unique variant trait associations that have been identified. And I highlight here on the lower side what I think some of the key impacts, the key factors that have been driving those discoveries. Many of them are technical, scientific, and also some of them are cultural. For example, the willingness to share data and participate in meta-analysis. The willingness of the UK government and medical systems to invest in UK Biobank and make those data openly available to all qualified researchers. So one can look at this and potentially opine, as has been done over the past 6 or 7 years. Everyone keeps asking, all right, have we reached the end of the GWAS? Have we found everything that there is to find? Is there any reason for us to be continuing to do these types of studies? And so if I look into my crystal ball and make some guesses as to, given where we are today, where we're likely to be over the next 10 years, I personally feel like we have yet to hit the inflection point in terms of discovering how genes influence risk of disease, our response to disease, the severity of the disease that we have, and the trajectories that we follow when we get those diseases. And each one of those new discoveries could point to potential new therapeutics for drugs that need to be developed. So what has been our grist for finding new genetic associations? Well, in the past, it has been primarily public databases. So, unless you were Regeneron or Amgen who bought Decode or a few other of select few, most of us were primarily either getting access to bespoke case control cohorts for traits that we were particularly interested in, or we were turning to the public databases that were available. We also tried leveraging collaborations to be able to get access to potentially some unique case control cohorts or other groups that might provide unique input. But our experience with collaborations was that they were very time-consuming to establish. And usually by the time we got an agreement signed, analyses run, and data back, 2 years had passed, and we were no longer even interested in the particular area that we had set out to establish the collaboration for. As Alan indicated, GSK, Merck, and many other pharmaceutical companies also collect DNA for a majority of our clinical trial participants. And a lot of us have a fairly routine approach to whole genome genotyping those clinical trial participants and analyzing them primarily from a pharmacogenetics perspective to be able to assess whether the variants are having any effect on drug efficacy or could explain safety events that we're seeing. But there's also, we're at least internally, getting more and more interested in the detailed clinical information that we're collecting on some of these trials. At GSK, we now have over 100,000 individuals that have come through our clinical trials, and we have whole genome genotype array data for. Many of those are diseases or disease subtypes that we continue to be interested in, where we don't feel like we've learned everything that we can, and there are some, at least limited opportunities to use those data for new discovery. Biobank is certainly the game changer, and I think over the next 5 days we're going to hear plenty about the value of UK Biobank. There are other real-world data-linked genetic data sources that are emerging. Many of them that are publicly funded, some of them that are not. But I think over time, the same trends that we've seen in the broader case control GWAS field will probably bear out in the EHR-linked data fields as well, and that is mechanisms for data sharing. And GSK has also made other investments in that area. I'll say a word or two in a couple of slides about our collaboration with 23andMe and our soon to have involvement on FinGen as well, a Finnish biobank. Ultimately, I think in the coming years, the next game changer is going to be the ability to carry out these cross-biobank analyses, where each biobank may be from 50,000 to 500,000 or 1 million participants. If you bring 30, 40, 50 of those resources together, the opportunity for novel discovery and exploring the genetic effects in very particular patient subsets could be very powerful. I'll say a few things about some work that we're doing in UK Biobank. Like I said, UK Biobank was a complete game changer for us from how we can go about identifying genetic effects and characterizing them quite extensively across a wide range of traits and diseases that are collected in that excellent resource. This highlights just 4 major areas of research that we've kind of binned this into. So PGRN for every target. So you could imagine that any target that we already have a drug in development for or that we're working on, we want to try to understand what does the genetic data tell us about, are we looking at the right indication? Are we looking at the right patient subpopulation? And are we at risk for any potential safety concerns? And so that's an active area of interest for us. Conducting genome-wide analysis for key traits. We've defined approximately 2,500 traits for UK Biobank, where we have developed phenotypes for them and are just about have completed the process of carrying out all of those GWAS with both logistic and linear models. Diversifying the phenotypes, I mean, that is the beauty of having access to a subject-level research cohort like UK Biobank, where you can get access to a very rich set of data, including continuing to grow electronic health record data from the hospital episode statistics, and hopefully soon to have the general practitioner data on at least half of UK Biobank. And so there's just a constant activity around examining and re-examining the way that we've defined those traits and asking, can we better understand a particular patient subset and the genetic effects there that might help to select a target. And then exome sequencing. GSK and Regeneron a year and a half ago entered into a collaboration to sequence 50,000 exomes from UK Biobank, and Regeneron is continuing with other partners to sequence the remaining 450,000. We did receive the first 50,000 exomes back in June, and Regeneron and GSK are both in the process of conducting those analyses. Those sequence data will be available after 9 months, so in March 2019, they should be, the data will be unembargoed. Whether you'll actually be able to download them at that time is always a crapshoot. So what are some of the studies that we have been doing? I'll just give a couple of examples with UK Biobank. So one of the first that we carried out was osteoarthritis. Osteoarthritis is kind of low-hanging fruit because it is a relatively common condition. It is pretty well captured in UK Biobank, both through questionnaire data as well as through some of the electronic health record data that's available in UK Biobank. We defined 4 different traits for osteoarthritis for this analysis. So we had knee OA, hip OA, knee or hip, or any OA, any joint being affected. And so this, because it's a relatively common condition, the UK Biobank had roughly 4 times as many cases as the largest GWAS that had previously been published. And so as you can imagine, this was a ripe area for discovering novel loci. So we identified across those 4 analyses 64 unique loci that were genome-wide significant. 52 of those loci were novel, not previously reported from the other GWAS. We went through a fairly extensive variant to gene mapping effort, that's the V2G, with a variety of methods to identify potentially 216 genes underlying those 64 associations. Some that we could map quite uniquely to a single gene with pretty high confidence, and many loci where we have multiple genes that we can't really disambiguate based on the available genetic data that we have. We're currently in the process of going through and of some of the genes and the mechanisms that seem to be most promising for OA. We're investigating them in a number of screens with tool molecules that we have available, as well as some gene editing activities there. If you're interested in this, there's one talk and one poster that's listed here at the bottom from my colleagues Linda McCarthy and Ioana Tachmazidou, who will go into this analysis in quite a bit more depth. So another example is asthma. Again, this is another opportunity of low-hanging fruit in UK Biobank, because asthma, again, is another pretty prevalent condition. I think there are about 60,000 subjects in UK Biobank that report having asthma. There are a lot of very effective therapies out there for treating asthma, but exactly which asthma patient subpopulations you may want to go after and what untapped mechanisms might be out there to address some of the patient populations that don't respond particularly well. So in this study, we had interest in trying to understand what differentiated the risk factors, the genetic risk factors for adult versus juvenile onset asthma. So my colleague Ki-Jung Sung, who's also presenting on this on Thursday, identified those that were clearly very early childhood onset, 12 years or younger, of which there were 11,000 cases, an adult onset later than 40 years, with 17,000 cases, with an independent set of controls for the two. And as you can see here, that, and perhaps not surprisingly, that we found much stronger genetic predisposition and far more genetic associations that were genome-wide significant for the childhood onset. And in the figure on the right shows the test of heterogeneity between the childhood onset and adult onset. And you can see that the risk factors underlying the two are quite different. So UK Biobank is fantastic, especially for studying quantitative traits and conditions that are relatively common, but you don't have to go too far down that disease prevalence spectrum before you start getting into lots of things that we're interested in that it's not particularly well powered for. So as it turns out, asthma and osteoarthritis are probably two of the best powered GWAS that one could carry out in UK Biobank. This is sample size on the bottom in millions. And statistical power for the condition that I have there in the lower right-hand side on the y-axis. And so even things that are still relatively common and of high interest, such as migraine or osteoporosis, in 500,000 individuals from a population sample like approximate population sample like UK Biobank, the power tapers off there very quickly. So you really need much larger data sets if you want to use this type of resource for investigating the genetics, the genetic risk factors involved. Not to mention if you're potentially interested in subsets of those diseases that might be defined by other factors. So clearly, there's a lot of opportunity in the future as these types of resources grow and as we can integrate data across resources like UK Biobank to be able to drive discovery in far more areas. So it brings us to a collaboration that we announced back in July between 23andMe and GSK. And it was exactly the observations in the previous slide that are heavily driving this collaboration. 23andMe is a direct-to-consumer genetics genotyping service. They've been around since 2006. They have kind of built themselves as a customer-friendly platform, providing back to their customers a lot of information about that they are able to provide about their genetics and implications on their health status and traits. It's a fairly Northern European-centric database at this time, which is predominantly European, but growing in other areas, including African Americans and Latinos. But really what is most remarkable is the growth curve in 23andMe. They have essentially doubled in size in each of the last 3 years. They're currently just over 5 million customers in their database. Over 80% of those customers have consented to participate in research, and so they provide responses, self-report responses to questionnaires, which allows us to delve into the genetics of a wide range of different types of traits and characteristics. And if the pattern continues, then we could anticipate that that resource could be 6 million next year, 12 million the following year, and continue to grow from there. And even though some of these minority groups, such as African Americans or customers of African descent, are a relatively small portion of the database as a whole, 5% of 5 million is the single largest African American collection with genetic data in the world. And so it is a tremendous opportunity to be able to explore the genetics of important characteristics. Also, their ability to interact and engage their customer base quite fluidly and often in phenotype or genotype directed manners allows them to deploy new surveys to those customers to collect new information. And so if there is a particular subtype of a disease that we're interested in that wasn't envisioned when the existing sets of questionnaires were developed, new questionnaires can be quickly developed and deployed to be able to fill in those holes. So it's a fantastic opportunity to grow our body of those genetic associations that fuel this research. So I'm going to digress just a little bit and kind of speak back to the title that I was assigned for this, and that's the relative role of sequencing and whole genome genotype arrays in this type of research. I think there's definitely a lot of room for debate and discussion about this, and there's plenty of value that's been demonstrated through scientific papers for both array-based genotyping and sequencing. And I think Alan did a beautiful job highlighting where sequencing has a particular role in these founder populations and consanguineous populations, where you have an opportunity to identify these variants that have been enriched within particular families or populations. You know, the real argument about, do we sequence or do we genotype I think ultimately, unless you're dealing with a special population, it comes down to bang for your buck. So kind of based off of large-scale costs that one can get to these days, you can, for $10 million of available budget, you can whole genome genotype 200,000 individuals. You could exome sequence roughly 33,000 individuals, or you can genome sequence, whole genome sequence around 12,500 individuals. And so if your intent is to use those data for genetic discovery, then the statistical power you will have to make any value out of those data are going to be quite a bit higher for the large numbers that you could whole genome genotype than the smaller numbers that you could genome sequence. And if you try to assess this just from the vantage point of, well, so what are you capturing when you genotype versus when you sequence, and when you exome sequence versus when you whole genome sequence? And I've tried to capture this in the figure here on the right. Whole genome sequencing gives you relatively uniform capture across the entire spectrum of allele frequency, which is shown on the x-axis here. Whether you're in the exome or in the rest of the whole genome. Exome sequence, of course, does a really good job in the exome, but captures very little outside of the exonic regions. And in fact, whole genome sequencing, at least has historically done a somewhat better job of giving you uniform exome capture compared to pull-down methods for exome sequencing. And then in blue, we have the kind of the current power, or in this case coverage, of the array data from UK Biobank. And so this is based off of imputation using the UK 10K and the 1000 Genomes Project data. And so it's really great if you're talking about common variants. You're essentially capturing nearly all of the common variation at 5% frequency or greater. It's pretty good in the low frequency range at 0.5% to 5%. And then drops off as you get below 0.5%, and of course, below 0.05, it's the ultra rare spectrum. It's quite poor. Now, the coverage is for imputation is almost entirely dictated by the total number of minor alleles that you observe in your data set. And so, as the body of whole genome sequence data that can be used for training those imputation panels grows, for every 10-fold increase in your imputation data set increase, you're going to be able to impute at roughly the same coverage 10-fold lower allele frequency. So when we get up into the range of having about 1 million whole genome sequences that we can use for our imputation panels, we'll be at a point where we can impute with very high power down into the 0.01, 0.001 minor allele frequency range. And then just to talk about this in terms of the capture of the number of genes for which you would have over 80% power based on the cumulative frequency of loss of function variants in those genes, comparing the genotype array versus the exomes, going from left to right within each of those odds ratio bins from the 450,000 genotype arrays to 50,000 exomes, 200,000 exomes, or 450,000 exomes. If the effects of those loss of functions is relatively modest, exomes will almost, or whole genomes, will almost never exceed by much the power of genotyping in terms of the number of genes where you can identify those. But where you have much larger effects of those loss of functions, you essentially at roughly a 2-to-1 ratio, you get in terms of the genes with sufficient power to identify those large effects in the sequenced versus array genotyped. And again, I won't dwell on this, but this is essentially the argument that was made. And this is a great paper that illustrates the value of sequencing these special populations like PROMIS or the Pakistani Genome Resource, and showing your ability to capture individuals that are homozygous for loss of function variants, so that you can go in and carry out those follow-up studies and assess what the impact of being a complete knockout for a gene is in humans. So understanding genetic impacts, I've already spoken to this to some degree. This is one example. So IL-33, there was a paper published a year and a half ago by Decode showing that there was a low frequency, 0.5% frequency loss of function variant in IL-33. IL-33 is a drug target for asthma and a number of asthma-related conditions. GSK has 2 drugs, one in the clinic and one entering preclinical testing for IL-33 receptor and IL-33. And so these are some of the kinds of questions that we can go and ask in UK Biobank. What is the effect of that variant in all comers of asthma, or asthma with low eosinophils or high eosinophils, childhood asthma versus adult asthma? And of course, we see the bigger effect in the childhood asthma. We also see it significantly associated in COPD, hay fever, nasal polyps, but not associated with all reasons for death. We can look at this in a different way. So this is doing a PGRN across all 2,500 traits that we've conducted these analyses in UK Biobank for. And quite reassuringly, we're essentially seeing all of these asthma and respiratory related phenotypes that are coming up through the PGRN, and no other potential safety hazards being highlighted through this analysis. So it seems to be quite specific. So that was with the loss of function. I'm now showing the results for a relatively common variant in IL-33 that's associated with asthma, and it's an EQTL. And we see essentially the exact same pattern. And where there are relatively few genes where we have these types of loss of function variants, like we have in IL-33 to study them, we have, as we found, over 17,000 genes for which we have an EQTL like this, where we can go through and essentially instrument those genes and assess the impacts of those genes. And comparing these two slides, we see nearly identical effects between the loss of function and the EQTL. So variant to gene mapping, there are a lot of different methods and data sources that we can apply to this. I'm going to just talk about a couple. I mean, first of all, is the importance of really understanding your region of association when you're dealing with high-density genotyping like this. So this is that IL-33 locus that we were just talking about, where you've got that very high peak for the EQTL variant that's shown, and you've got the shoulder down there for that loss of function. Being able to go through and do a conditional analysis in each of these regions to identify each of the conditionally independent variants is a critical component to this variant to gene mapping. In this case, having done that conditional analysis and finding that a loss of function in IL-33 would provide pretty strong evidence that more common variant was also acting through IL-33. An important advancement in terms of our methodology for identifying using EQTL or protein QTL data for mapping variant to gene is this concept of co-localization. If you've looked at any of these PGRN type plots and you pick a variant that you like because it has some effect that you're aware of, and you look at it across a number of different traits, you might find that it's significantly associated with a number of different traits. But if you were to drill into the region of each of those different associations, you might find that the peak of that association is far away from the variant that you're finding that significant association in your PGRN. That is, that association is not co-localizing with the association or the variant of interest. And this is just one way that we have come up with to help illustrate that. So a good example is HMG-CoA reductase. And in HMG-CoA reductase, if you take the peak SNP that's associated with that for LDL cholesterol, and you do a PGRN, you see that it comes up with a wide range of body fat and weight-related traits that are associated. But if you conduct a co-localization analysis against essentially all tissues and all genes within those regions, for each one of those genetic associations, you find that the cholesterol-related associations all co-localize with HMG-CoA reductase, and actually this COL4A3BP, and that all of the body weight associations co-localize with the ankyrin gene here on the right. So if you're only looking at your PGRN plot, you're going to be misled that your gene, HMG-CoA reductase, is also potentially beneficial for weight loss, which in fact it is not. So I'm going to go ahead and conclude there, and I'd like to thank my colleagues that contributed to this work. And as I showed on the slides, several of them are speaking in this conference if you'd like to learn more about this work.
Hi, Julie Lynch, Department of Veterans Affairs. I just wanted to make a comment about relying on UK Biobank and even your clinical trial databases, is that there aren't enough African Americans, particularly with disease. And the VA Million Veteran Program has 20% African American with, high number of disease. And so I think it's really important that we develop biorepositories that are adequately representative of minorities.
Yeah, I couldn't agree more. And I mean, for that reason, many of us are very interested in collaborating with Million Veterans.
So that's a very good point, and I just sent my colleagues an email about this. And being able to efficiently collaborate with industry, I think, is a big problem that needs to be resolved within the VA for this.
Yeah, it's one of those that we've tried many times, and it's challenging. I think the only thing I'd say is that I think, in quality, it's very similar to what you'd find in the Regeneron Discover publication. I think that the observations are, that's exome sequencing, a population sample like that is kind of, those are the kinds of observations that come through. I don't think that we had high hopes that we would find many screaming signals, say, for particular diseases in that. And so, I mean, that's, I think it's about what one would expect for those types of data. Okay, thank you.